Now let's talk about taking our single-stranded polynucleotide of DNA and how it can actually form pairs between bases using hydrogen bonds. So between two bases of nucleotides, we can actually form a hydrogen bond pair. Now, for a polynucleotide, this type of pairing is restricted essentially to two types of pairing. A will pair with T in DNA, and C will pair with G. Now, of course, when we're talking about RNA, if you do have any pairing going on, the T here is, of course, replaced with a U. Essentially, the idea is that purines will pair with pyrimidines, right? We have our purines and pyrimidines, which are the separate types that make up these pairing units, A and T and C and G. So again, A pairs with T, C pairs with G. How does this occur? Well, first, let's go over some differences in content between A and T and C and G when looking at our DNA. So based on what we just discussed, that A nucleotides can pair with T nucleotides and G can pair with C nucleotides, in something like double-stranded DNA, you have two strands, and down those strands, of course, you might have your nucleotides like this. And we then know that these will only pair with the opposite, which we just talked about. A only pairs with T, T with A, C with G, G with C. Because of this structure, the percentages of A and T and C and G in a strand of DNA can easily be distinguished. We know that the percent of A that is in a DNA strand will be about equal to the percent of T that is in a DNA strand. Similarly, the percent of G should be equal to the percent of C. And this is pretty validated within anything that has double-stranded DNA. This is validated by this Watson-Crick pairing, A and T, C and G, in the model. Now, within one strand of DNA, generally, it still does hold true that the amount of A present is about equal to the amount of T present, and the amount of G present is equal to the amount of C present. Now, when we just talk about a single strand of DNA, the reason that this still kind of holds true is because that single strand of DNA usually needs its pair strand, which has to have the equivalent amount. Something like RNA, which does not bind in a fashion where A and T and G and C are going together, doesn't necessarily have to have a matching content of A and T and C and G. And again, that's just because there's no second strand there that is matching our pairs. This can actually be seen within organisms as far as the full content of DNA when we measure something called the GC content. GC content is just a measurement of how much G and C exists. Now, G and C pair together, but that doesn't mean that there can't be just 40% or 20% GC. G and C happens to be a stronger pairing bond. And so DNA that has less GC content is usually thought of as being less stable than DNA with high GC content. When we look at the average amount of GC content in a human, this ranges from 35 to 60, usually around 50%. Now, in other organisms, the GC content is different. So, for example, something like yeast only has about 38% GC. That means that the rest of it is A and T. Another organism, the Thale cress, has about 36%. These are other eukaryotic type cells with DNA, but again, they have lower GC content. However, there are some organisms that utilize this high GC content, again, because it stabilizes the DNA, it makes DNA more stable. So, for example, actinobacteria, bacteria that are usually categorized as potentially being in harsher environments, have a very high GC content. So one example of this is Streptomyces colicolor, which has a GC content of about 72%. Here is the actual hydrogen bonding that occurs between AT and GC pairs in a DNA type structure, or a double strand of nucleic acids. You can see that adenine, which is shown here, and we're showing right here is where it is bound off to its sugar on the side. Similarly, thymine is bound off to its sugar right here. You can see using the 6-membered ring of adenine and thymine, we are making 2 hydrogen bonds. So A and T only bind together with 2 hydrogen bonds. This is a hydrogen oxygen bond, so the carbonyl on our thymine and the hydrogen off of an amine group on an adenine. And then also a hydrogen nitrogen bond. So again, 2 bonds, and of 2 different varieties. They're both hydrogen bonds, but one is a oxygen-hydrogen type hydrogen bond, and the other is an a nitrogen-hydrogen type hydrogen bond. Now let's move to visualizing the guanine cytosine hydrogen bonding. You can see that the binding between these two is similar in nature. Again, our cytosine, which is the other purine in this case, is utilizing a hydrogen off of an amine group to bind with the carbonyl oxygen on the guanine. So we have an oxygen-hydrogen bond. Here, another amine nitrogen, hydrogen off of a nitrogen binding with a nitrogen group. This is a nitrogen-hydrogen bond. But we also have a third type interaction where we have a hydrogen interacting with another carbonyl, making another hydrogen-oxygen type hydrogen bond. So again, here, oxygen-hydrogen, nitrogen-hydrogen, oxygen-hydrogen. This is where the stability of the GC type bond comes in, because there are 3 hydrogen bonds connecting it, giving it higher stability than the AT type interaction. Now, I want you to think about why A and T can't form a third hydrogen bond. Look at our hydrogen bonding that's occurring here in guanine and cytosine on our bottom position where we have an amine hydrogen binding with that carbonyl here. Note that thymine also has a carbonyl at the bottom, carbonyl oxygen. And why can't it bond with this hydrogen? This is a question I'm going to pose to you to think about. Think about how hydrogen bonds must form and the characteristics of the hydrogens that need to be present for a hydrogen bond.